There are mainly 3 research directions going on in the lab. First of all, we would like to use genome-wide CRISPR screen to discover novel regulators in macrophage function. In this particular project, we developed a genome-wide CRISPR screen for macrophage efferocytosis, and I'm going to share a story about a gene we found from the screen WDFY3 as a novel regulator for efferocytosis by macrophage. Another research direction is starting from human genetic discovery. We identified the candidate risk loci associated with human coronary heart disease and try to interrogate the mechanisms, including the causal variant. In that particular case, we are interested in a gene called lysosomal acid lipase, LIPA, a risk locus for human coronary heart disease. We would like to study its role in atherosclerosis. And this is the project funded by my K99 and R00 grant. Another direction is we established and published a protocol to differentiate human-induced pluripotent stem cells to macrophages. We use this protocol to study human macrophage-specific biology, for example, human-specific long non-coding RNAs, human genetic variants discovered by exome sequencing for Mendelian diseases, and GWAS. And for today, I would like to mainly talk about this project. The rationale is why we are interested in macrophage efferocytosis and why we think it's important to use CRISPR screen to study macrophage efferocytosis by discovering novel regulators in an unbiased way. And what we have found by doing a CRISPR screen in macrophage for efferocytosis phenotype. So my talk is gonna cover all the 3 questions. First of all, why we are interested in macrophage efferocytosis, right? So efferocytosis is referred to phagocytosis of apoptotic cells by phagocytes, which include macrophage and mainly by macrophage, but also involve other cell types. We are interested in efferocytosis because macrophage efferocytosis is very critical in maintaining tissue homeostasis. A healthy adult will turn around over 100 billion apoptotic cells in a daily basis. These cells will need to be cleared away, otherwise they will go through necrosis, trigger inflammation. Defective efferocytosis indeed drives important diseases, including cancer, autoimmune diseases, and atherosclerosis. And then therapeutics to enhance efferocytosis is beneficial. So one example is that the anti-CD47 antibody that improved the cancer prognosis by enhancing macrophage efferocytosis of cancer cells has been successful and now is being evaluated in phase 2 clinical trials. Which is a big thing in the field because it's basically suggests that this pathway is therapeutically targetable. And for atherosclerosis, there are numerous experimental studies to support that enhancing efferocytosis is protective against atherosclerosis in animal models. And in human atherosclerotic plaques, there is clear evidence supported by seminal histological analysis that efferocytosis is impaired. So basically, we think macrophage efferocytosis is a fundamental biological function performed by macrophage and has important implications in multiple diseases and is a therapeutically targetable pathway. We actually have learned a lot about how phagocytes perform efferocytosis. So basically, the phagocytes needs to recognize apoptotic cells and then bands them, internalize the apoptotic cells, and eventually degrade them so that the efferocyte can be ready for take up another apoptotic cells to continue the process in an efficient way. So far, a lot of the findings are derived from very productive and important hypothesis-driven studies, but the unbiased approach has also played a very important role. We learned a lot of things from the previously performed unbiased screening to really gain a big unbiased view of what are the regulators for this biological process. For example, in very early studies, genetic screen has been performed in non-mammalian macrophages for efferocytosis regulators. And there are also studies to look at the proteomics of the isolated phagosome, basically to search for proteins that are important for phagosome formation. And CRISPR screen has also recently been performed in human monocytic cell line-derived macrophages to identify regulators for phagocytosis of bees, xylosome, and myelin, different substrates. And indeed found that there's a substrate-specific regulator for the phagocytosis process. However, we think it's important to perform a CRISPR screen in primary macrophages using apoptotic cells as the substrates, because it is apparent that the monocytic cell line derived macrophages are not physiological. We actually found that these cells showed a pretty poor efferocytosis capacity. So if primary macrophage, after you feed them with apoptotic cells, can demonstrate over 30% of the macrophage that actually engulf the apoptotic cells, we usually only observe like 2% for tumor-derived macrophages. Therefore, they may perform efferocytosis through different mechanisms. And because of the substrate-specific mechanism, including recognition, binding, internalization, and degradation of apoptotic cells, which could obviously be different from engulfment of bees or other substrates. We think this is the gap in the field. Therefore, for this project, we decided to establish a CRISPR screen in primary macrophage and using apoptotic cells as the substrate. So to develop a novel screen in primary cells, which is typically not done in the past, and then for functional phenotype that beyond some relatively simple phenotype like biomarker expression or cell variability, there are some technical challenges we had to overcome. That mainly includes statistical power. We need to have a lot of cells to be able to maintain the genome-wide coverage. And editing efficiency in primary cells, because it's a lot harder to reach ideal editing efficiency in primary cells compared with tumor cell line. And then also for the enrich method of the phenotype, we need to be able to have a realistic protocol to be able to separate the cells that can perform efferocytosis and those cannot. In that way, we can sequence them and then calculate the enrichment, identify the highly ranked potential candidate genes. So with this in mind, the goal of this project is to establish a novel screen to map regulators of efferocytosis in primary macrophage in an unbiased manner on genome-wide scale. And of course, to discover, hopefully, the novel regulators of efferocytosis and review novel mechanism and potentially therapeutic targets. So we were able to do it. Here is the schematic for our screen. We used a genome-wide CRISPR library called Brie library that has 80,000 guide RNAs that targets 20,000 genes. And we used the bone marrow cells from Cas9 transgenic mice. So those cells already had a stable expression of Cas9, and we only need to introduce the guide RNA. We used the lentivirus to transduce the guide RNA into the bone marrow cells and maintain a low transduction ratio so that majority of the bone marrow cells only got one guide RNA, which will edit 1 gene per cell to minimize confounding effects. After the editing, we differentiated bone marrow cells into macrophages And then we performed so-called two-round efferocytosis to try to enrich those macrophages that are very good at eating and those did not eat. So basically, we used two different fluorescence probes to label the apoptotic cells. And the first round, we used a green fluorescence labeled apoptotic cells and allow the macrophage to eat, and then wash them away and add a red fluorescence labeled apoptotic cells. So in the end, you can imagine that if macrophages are very good at eating, and they will be able to engulf in both rounds and show positive fluorescence for both red and green. And those poor eaters did not get to take apoptotic cells, even given them two opportunities, will remain negative for both fluorescence. So the flow cytometry panel will appear to be like here. So about 5% of the cells are the good eaters and engulf the two apoptotic cells, and around 35% of cells did not eat. In that way, we can actually enrich our phenotype. The next step is we isolate the genomic DNA that actually contains the guide RNA. So remember, the guide RNA was introduced by lentivirus. It will be incorporated into the genome and serve as a barcode to indicate which gene has been knocked out in that particular cells that lead to the phenotype of either supereater or non-eater. We would then sequence, count the guide RNA number and perform enrichment analysis. So the results kind of look like this. If we plot the count of guide RNAs in the good eaters versus the non-eaters, majority of the guide RNA would align along the diagonal, which means that they didn't do anything. They didn't get enriched in either population. But if you can imagine that if some of the genes are very important for efferocytosis, you knock it out, the macrophage cannot eat anymore. Those guide RNAs would be enriched in the non-eaters. And as clearly shown that the RPC4 and NCKP1L are the known regulators of phagocytosis and efferocytosis, and those guide RNAs are being enriched in the non-eater population. And the WDFY3 is one of the novel ones that has never been implicated for efferocytosis or regulation of phagocytosis. So this is our workflow, seem to be successful. Also because that the top positive regulators we were able to pull out from the screen clearly enriched in the known pathways involved in phagocytosis. For example, actin polymerization, Fc receptor mediated phagocytosis. The next question is about how we prioritize our screen hits for the mechanistic study. It wasn't too challenging because we can clearly find that the MAGIC and ScreenBeam, which are two independent algorithms for screen data analysis, both pulled out 3 genes ranked among the top 10 of the screen hits. And then WDFY3 is the novel one. So we began to be interested in this gene and we decided to validate. The method we validated our top hits is kind of similar to what we did with the screen, but in this time, we only edited our bone marrow-derived macrophages with guide RNA, either non-targeting, which serves as a control, or that targets WDFY3. And we would label apoptotic cells, which I would use AC to refer to it in my presentation. We will label the apoptotic cells AC with a red fluorescence and feed them to the bone marrow macrophages being edited. And then some will take, some will not. And in the end, our readout is a percentage of the macrophage that indeed engulf apoptotic cells. So for the rigorous of the research, we tried 2 independent guide RNAs targeting WDFY3. And both of them showed that impaired efferocytosis were observed with knockout of WDFY3, as shown here and here. And we used different ratio of apoptotic cells with bone marrow macrophage to give the macrophage increased challenging that is so-called high burden efferocytosis so that we can further review the defects. And it is clear that the knockout of the WDFY3 appears to suppress efferocytosis by over 50% if compared with the non-targeting control. So what we have known about WDFY3? It is a gene highly conserved, mostly abundantly expressed in neurons. And it was known to be required for autophagic clearance of aggregated proteins by binding with autophagy machinery with their known protein functional domains. So this leads us to hypothesize that WDFY3 may be involved in the regulation of apoptotic cell degradation after engulfment, in addition to the uptake, which apparently was able to validate. So we used, in that case, because whole body WDFY3 knockout is embryonically lethal, well, we decided to use a tamoxifen-induced knockout of WDFY3 and then isolate bone marrow macrophage. So here we show that the efferocytosis of the inducible knockout mice bone marrow macrophage were also compromised, although to a less extent compared with the transient knockout, which induced almost over 50%. But the more obvious phenotype we were able to observe is that the inducible knockout mice macrophage showed impaired degradation of the apoptotic cells, which is shown here that after the engulfment and allow them time to rest and do their job, then we see that in a normal macrophage, the apoptotic cell fluorescence appear to be fragmented. But when there is defects in degradation, we expected to see that engulfed apoptotic cells remain undegraded. So this is how we analyzed and draw the conclusion. Our hypothesis is that maybe WDFY3 participated in the phagosome formation based on its known binding site with the autophagy machinery. And we try to do this experiment in human macrophage for human translation. So we got pretty successful knockdown, particularly at 72 hours. And we were able to see that in human monocyte-derived macrophages, siRNA-mediated knockdown and impaired efferocytosis. And then immunohistochemical staining shows that WDFY3 is cytoplasmic localized and appear to be co-localized with engulfed apoptotic cells, which is labeled red here. So we know that immunohistochemical staining could be not specific. So we took another approach. We used CRISPR editing to generate the cell line of THP1 derived macrophage with the insertion of a GFP tag fused with the C terminals of WDFY3. So the fluorescence shows the endogenous expression of the WDFY3. And this is lab cell imaging, so no nuclear staining, but it's apparent that WDFY3 is cytoplasmic localized, and this is a bees, and appears that WDFY3 is co-localized with it. But it is also obvious that like the beads is so much smaller than the apoptotic cells. And also because the previous screen using beads did not really pull out WDFY3, we think it's important actually to use apoptotic cells to study the role of WDFY3. So we're gonna try it again. This is also highlight why we need to use primary macrophage, because the THP1 macrophage is just so poor at efferocytosis, and we tried and we could not even able to find a macrophage that engulfed one by lab cell imaging. But using this cell line, we were able to draw another conclusion, which we found is interesting, is by feeding the THP1 macrophage with apoptotic cells, over 24 hours, and we saw the GLP fluorescence are upregulated with the apoptotic cell stimulation, which we think that WDFY3 must be required for the efferocytosis process that is upregulated. And then what could be the potential molecular mechanism deep down, right? So the good thing is literature have already.
Anne-Marie, can I just interrupt you? It seems like you're not sharing your screen anymore.
Oh, okay.
So I don't know if something changed with the settings.
Let me
but
try again.
Let's see if.
My part still says host disabled. Screen
Here we go.
sharing.
Hang on a second. Let me
Okay.
see. Try that. Try that. There we go.
Sounds good.
Start again. Or not start again. Start from the beginning of this slide.
Okay, this slide or this slide?
The next slide. Yeah.
Okay,
yeah.
perfect.
Yeah.
Yeah, nothing got missed. So you.
Yeah. The next slide. Yeah.
Perfect. Yeah, some previous literature has already pointed out that WDFY3 is directly interacted with some of the important autophagy machineries, including RepGap1, L, and LC3. So the gene symbol is not that easy to recognize, but this is LC3. So WDFY3 may be participating in the LC3 associated phagocytosis. And then what about at the transcriptome level? We are still looking into this data deeper, but it seems like there are not a lot of differentially expressed genes when we perform RNA-seq of bone marrow-derived macrophage in control and inducible knockout mice. But with the current data, it seems like higher lysosomal genes are expressed in the control cells. And then genes related in TGF-beta signaling are higher in the inducible knockout macrophage. So it appears that the protein may regulate lysophagosome degradation through interaction with the autophagic machinery. But at RNA level, the knockout WDFY3 might affect a different set of genes. Obviously not any of the autophagic genes we could ever find from our RNA-seq data. So it's like a transcriptional level and protein binding level analysis independently. We also try to determine whether WDFY3 is required for efferocytosis in vivo. We use bone marrow transplantation of lysozyme negative or positive WDFY3 flux mice to induce myeloid-specific knockout of WDFY3. And then after the reconstitution, we give the mice either PBS or dexamethasone to induce apoptosis in the thymus. And then we isolate thymus, and our readout is the percentage of the apoptotic cells per thymus. And you would imagine that if efferocytosis is impaired, the percentage of apoptotic cells should be higher. The bone marrow transplantation was pretty successful. We see clear knockout of WDFY3 in the macrophage. And then after giving dexamethasone, apoptotic cell percentage are higher in the thymus in both groups, but it's further increased with the knockout of WDFY3. So we think WDFY3 is regulating efferocytosis in vivo. What about the role of WDFY3 in atherosclerosis? So we were able to see that among plaque macrophage, the WDFY3 appear to be enriched in a certain subpopulation of macrophage that are also showing enriched expression of M2 markers. And this WDFY3 expressing macrophages show the increased number during plaque regression compared with progression, suggesting that the WDFY3 expressing macrophage may be M2-like and increased during plaque progression, and supporting its potential protective role. And then by collaborating with the biobank, the biobank at Karolinski Institute, we were able to see that in human carotid and endarterectomy samples, the WDFY expression are positively correlated with a lot of the M2-like macrophage markers, again supporting that in human plaque, WDFY3 appear to be co-expressed with M2-like macrophage. And indeed, inflammatory stimulation seemed to reduce WDFY3. So we don't really have other data yet, but all those data give us confidence to go for the next step to study the role of WDFY3 expressing macrophage in atherosclerosis, which will be the next step. Another line of the research is that by collaborating with Saleheen, who established the Pakistan genomic resource, we were able to identify over 200 human coding variants in the WDFY3 gene. And then by performing a very preliminary vive analysis, we found that the top associated phenotype is like PEN1 walking, which is a phenotype for atherosclerotic peripheral artery disease. Again, seem to support the role of this gene in atherosclerosis related phenotype. What we are gonna do as the next step is we have our established iPS-derived macrophage model, and we were able to show that monocyte-derived macrophage, iPS-derived macrophage from the same subjects appear to express WDFY3 at a similar level, which give us the confidence to model the human coding variant in WDFY3 using iPS-derived macrophage. So we this is some work, ongoing work too. We try to obtain the isogenic line with or without the human mutation in order to study the consequence in human macrophage. So current work, we're gonna go deeper into the molecular mechanisms. I'm gonna start using myeloid-specific knockout and overexpression mice to review the role of WDFY3 in atherosclerosis. And we also have other interesting novel regulators pulled out by the screen. So we're gonna get busy to see whether we can find some additional interesting stories out of there. And then, about this study, we really think the CRISPR screen in primary macrophage for efferocytosis is able to discover novel regulators and pathways and can help us to review novel mechanisms and potentially therapeutic targets. And we also hope that we establish this platform for a functional genome-wide screen in primary macrophage for the field of macrophage biology. In the end of acknowledgement, this project is mainly led by Jian and Xun, and all the other lab members contributing to this project. I really appreciate their dedication and their trust in my mentorship. It's been a very productive team so far. And then this project benefits a lot from the very productive collaboration with world-leading experts, including WDFY3, human genomics, and obviously for atherosclerosis and efferocytosis, and also other experts in CRISPR screen. And I just received my notice of award for my R1 on this project literally today, so that was great. And thanks for the other funding support to my postdoctoral fellows in the lab. And in the end, some visual fun part, and this is the people in our cardiometabolic genomics program. So it's been a really fun team to work with. All right, thank you very much.
Okay, Henry, thank you so much. So we'll take questions. Just like last week, there should be a raise your hand function at the bottom of your screen. If you can raise your hand, and then we'll give you the chance to ask a question.
Because I'm a co-host, I can't raise my hand, so I'm like literally raising my hand.
Okay,
and because I'm a co-host, I get to ask first because you know I'm exerting my power. Henry, that was amazing, and the work is so wonderful, and I think your approach was very novel and definitely cutting edge, and then that's congratulations on your R1. I had a question about whether or not you kind of showed some co-localization data with, you know, the expression of WDFY3, is that right? WDFY3.
Yeah.
In the apoptotic cell in an individual macrophage. But I'm wondering if you've looked in, say, an atherosclerotic plaque from, you know, MERTK knockouts or some other versions of disabled efferocytosis or defective efferocytosis to see where the endogenous levels or localization of WDFY3 might be in those scenarios, in enhanced efferocytosis or disabled efferocytosis. Have you looked at that?
Yeah, I think that would be really important to look at it indeed, but because WDFY3 is a really huge protein, so the staining approach is not working very well in tissue. In cells, kind of okay, but in tissue, like we were not able to get any success. The good thing is now we have the transgenic mice with our expression of flag-tagged WDFY3. So in that way, once we breed those mice, hopefully we'll be able to really locate the endogenous WDFY3 in atherosclerotic plaque, at least in mice. For human plaque, maybe we can stain for RNA, but it's different from protein. So this is the technical challenge indeed, but I agree that will be really important to look at the localization in the plaque. Yeah. Mm. So I guess, I mean, for the single cell RNA-seq data, it's actually quite supportive that WDFY3 is enriched in a subpopulation of macrophage in a pretty specific way. So this, to some extent, supported their presence in the plaque in both human and mice. Mm.
Okay, next question is Ada. Yeah, please go for it.
Thanks so much, Henry. That was fantastic, and congratulations on your grant.
Thank you.
I was wondering, I had a technical question actually about your screen. I was wondering why you were doing the knockdown before the differentiation to BMDMs.
Right. This is critical technical question. So this is mainly because once the bone marrow cells become macrophages, it becomes very refractory for any genetic editing. This is just a macrophage machinery. It doesn't really let lentivirus to get into its genome. So the editing is much more efficient at bone marrow progenitor cell level. And then after the knockout, you can imagine that it's at the DNA level. It will take some time to actually affect the mRNA and protein level. And we found that for CRISPR gene editing, that time window is around 6 to 7 days, which means that after the gene editing, we need to allow for about 6 to 7 days for the editing to actually take effects at protein level. And because of that, it's also the best design to do the gene editing at the progenitor level and then wait until them to become macrophage to test for the phenotype. Yeah.
Okay, Esther, you have a question, I guess? Please unmute yourself.
Hi, beautiful talk. I had a quick question. Your selection is based on the super efferocytotic macrophages and the non-eaters. What happens in the in-between population? As, for example, in the atherosclerotic plaque, you mostly will have the in-between ones or the bad eaters. Did you had the chance to have a look at their profile?
For the screen pipeline, the way to enrich the super eater and non-eater is mainly to increase the statistical power. Because otherwise, if we separate by 50% eater and non-eater, that actually wouldn't help to have the calculation to fund the enrichment. So that was purely because of the technical need for CRISPR screen. And in the plaque, I would say based on the previous literature, the macrophage really need to be very good eater because it's continuously being challenged by apoptotic cells. So this idea of continued efferocytosis is important for macrophage to maintain the homeostasis and protect atherosclerosis. Yeah, but in an in vivo setting, it's probably challenging to determine whether a macrophage can actually eat multiple apoptotic cells because we are looking at a static state instead of continue the process. Yeah.
Okay, we have one more question. It's Mie, right? Okay, go for it.
exciting project. My question pertains to, you mentioned that WDFY3 might be implicated in LC3 associated phagocytosis. So I was wondering if you observed any impairment of efferocytosis when you knocked out any other genes that are involved in this pathway, such as Rubicon?
My memory is that Rubicon by Jennifer Martinez's work, the knockout impaired efferocytosis. Yeah.
And in your system, did you observe that as well?
We haven't tried that actually, but I think it would be actually interesting to have double knockout and see whether there's additive effects. Yeah.
Okay, thank you.
Thank you.
Okay, Henry, in the chat function, I see two more questions. One of them is from Asma, is asking whether MERTK appeared on the screen results.
Yeah, this is a very important question. We actually didn't see MERTK, but I would say the screen is only addressing, it's only addressing part of the entire pathway involved in efferocytosis. For example, if with the knockout of one particular gene, then the other genes would upregulate it to compensate for it. We will not be able to pull out those cells. And another reason is that like a screen is always noisy. And there is also a possibility that the particular guide RNA targeting a certain gene, for example, maybe MERTK was not efficient enough. So there's always false positive, which can be easily ruled out. We don't worry about it. If we see a false positive, we cannot validate, then we forget about it. But there is also possibility for false negative, which means that we missed something. And so we'll never conclude that our screen identified every known important efferocytosis regulator. But we can claim that we identified something novel, which has not been discovered before. But indeed, when I looked at my screen results, we're like, where is MERTK? This is the first thing I was trying to look for. Yeah.
Okay, great. And there's another question. It's from Wendai, and he's asking whether WDFY3 affect LDL uptake by macrophages.
Yeah, this is something we haven't tracked yet. So if WDFY3 regulate the endocytosis process in general, probably my guess is it would. So in order to test whether it is a general regulator of phagocytosis, we actually need to try different substrates, which we are currently trying. But what I can say is that the previous screen using bees, myelin, or zymosome as a substrates and perform Genome-wide screen didn't pull out WDFY3. So my guess is that it's something specific to apoptotic cells, but of course we'll need to test that. But yeah, this is an important question.
Okay, Henry, that was a great talk. Thank you so much. And now it's going to be Esther is going to introduce our second speaker.
Okay, thank you everyone for still hanging on. I'm going to introduce the second speaker, who is Menno de Winter. He's from the Amsterdam University Medical Center. He's also my lab neighbor. We share our laboratories, and Menno is a real specialist in macrophage biology in the context of atherosclerosis. His main topics are macrophage metabolism and macrophage epigenetics, and I'm pretty sure he will give us a good overview and some latest results. So Menno, the floor is yours. Can you share your screen? Yeah.
Am I visible right now?
Yeah, we hear you and you're visible, so.
Perfect. Okay, thank you very much for the kind introduction, and thank you very much for allowing me to speak at this wonderful seminar series, which is really, really inspiring in these, well, sometimes difficult times. As they introduced me, I'm working on macrophages, and today I will be talking on some projects we are working on in the lab, which have more a focus on metabolic regulation of macrophages in relation to cardiovascular disease. Our key interest is atherosclerosis, atherosclerotic plaques, and as you can very nicely see in this slide, if you look at atherosclerotic plaques, you can find all types of immune cells. And my group is specifically focusing on monocytes and macrophages and how they regulate atherosclerotic plaque growth and factors like stability and clinically relevant parameters. This old picture from Peter Libby still very nicely introduces the function or the role that macrophages play in the atherosclerotic process. They migrate into the arterial wall through chemotactic processes where they differentiate, where monocytes differentiate into macrophages and start scavenging modified lipids to eventually become large lipid-laden foam cells. And in the context, during this process, production of inflammatory cytokines is important, tissue factor is produced, and of course, as already was discussed by my previous speaker, cell death and efferocytosis are also important processes in this. Macrophages come in many different flavors, and this picture from Florent Ginoux very nicely illustrates the heterogeneity that we have in macrophages in vivo. It shows that tissue macrophages throughout different organs are seeded during embryonic development, during different stages of embryonic development. And in inflammatory diseases, illustrated or depicted over here, like allergy or atherosclerosis, specific stimuli mediate the recruitment of monocytes from the circulation that also then differentiate into macrophages. And the combination of these tissue-specific signals that mainly drive the tissue macrophages and these additional or exogenous and endogenous stress signals, you get a whole wide range of different phenotypes of macrophages that all have different functions and contribute differently to disease. A few years ago, we started working on this heterogeneity, and then the field was still a bit more simple. And we worked on in the concept of M1 versus M2 macrophages. And I still believe it's a valuable reductionist tool to study macrophage function in relation to disease. And at that time, we started investigating pro-inflammatory or M1 macrophages and anti-inflammatory M2 macrophages in atherosclerosis. And what we could very nicely demonstrate around that time by using immunohistochemistry is that in human atherosclerotic plaques, there's a different spatial localization of macrophage subsets. As you can see over here by immunohistochemistry staining for different markers that detect M1 macrophages or M2 macrophages, we could show that, for instance, the shoulder region, which is normally associated with a plaque rupture, contains more M1 macrophages and less M2 macrophages. While in the cap, there also seemed to be a dominance of M1 macrophages, but this difference was less clear. And finally, in the adventitia surrounding the atherosclerotic vessel, and where you see a lot of fat accumulation of adipose tissue, you hardly find any M1 macrophages, but this is mainly dominated by M2 macrophages. But of course, this M1 and M2 paradigm is simple, and there must be more heterogeneity. And I'm not going to talk in great detail about it today, but one of the approaches that we are taking and that many labs are taking currently is doing single cell analysis on human atherosclerotic plaques. And for this, we set up a collaboration with Gerard Pastekamp from Utrecht and Johan Kuiper from Leiden. And we are analyzing human atherosclerotic plaques and endarterectomy samples, which are collected upon surgery. And we harvest this, we keep a section that we will use for classification of lesions, that we will use for immunohistochemistry, and the remainder of the tissue samples are enzymatically digested and used for single cell sequencing. And by using this approach, and by gradually increasing the number of patients that we are analyzing, we were now able to demonstrate different populations that you find in these atherosclerotic plaques. And based on gene expression patterns, you can group, you can cluster these cells, and then examining the gene expression patterns in more detail and looking at marker genes, you can label them as well. And in this way, we can nicely demonstrate, for instance, large groups of T cells that we find in human atherosclerotic plaques, but also clusters of myeloid cells. And as said, I'm not going to talk about the details of this, and there you can talk I can talk about this for a very long time. But for this talk, I now zoom in a bit on the 3 macrophage clusters that we can identify using this approach. So we took the myeloid cells, we reclustered them, we threw out the dendritic cells that we saw, and we ended up with 3 macrophage populations that we call My0, My1, and My2. And when looking at different classical markers that are associating with macrophage phenotype, we could also link a sort of label to them, generating or labeling them into 3 different populations. First of all, the My0 population seems to be more on the pro-inflammatory side. It expresses, for instance, IL-1 beta, it expresses several caspases. The My1 population expresses also a bit of IL-1 beta, but is also, for instance, characterized by TLR4 expression and TNF expression, while the My2 population shows more absence of these pro-inflammatory markers and does express lipid-related genes like ABCA1 and ABCG1. These are which are classically associated with foam cell formation and which are driven by transcription factors like PPAR gamma and LXR. So so based on the immunohistochemistry analysis that we did a few years ago, and based on the single cell sequencing analysis, we can define different populations of macrophages, and they are either pro-inflammatory or they are more lipid driven. You could also call them anti-inflammatory because they express less of the inflammatory marker genes. And hereby, I think by the single cell sequencing, we can now define 3 populations, 2 pro-inflammatory and 1 more lipid-driven anti-inflammatory. In my group, we are now following up on these studies, and we are particularly interested in what drives these cells. So can we define, for instance, transcription factors that drive these different subtypes? Can we identify signaling pathways? Can we define, as some of you may know, I'm very interested in epigenetic pathways and epigenetic enzymes. Can we identify epigenetic enzymes that control these populations and that can be targeted as a therapy? But we're also working on metabolic processes that control these different populations. Oops. Yes, there we are. And today the focus is more on the metabolic aspects of these macrophages, and to briefly introduce metabolic regulations of macrophages and their phenotype, I put up this slide showing that again you have anti-inflammatory M2 macrophages and pro-inflammatory M1 macrophages. And the M1 macrophages, you can classically consider them to be more associated with increased glycolysis and, for instance, also increased activity of the pentose phosphate pathway, and less activity of mitochondrial oxidation, while the M2 macrophages more rely on these mitochondria, the TCA cycle, and oxidative phosphorylation. And of course, by now, what we don't know, we by now know that it is not a black and white difference that really determines these subpopulations, but it's still a good guideline. So we started a project where we wanted to investigate these metabolic characteristics in foam cells. And in human atherosclerotic lesions, you can define these foam cells, macrophage-derived foam cells, characterized by these foamy appearances. And in mice, it's often more easy to see. This is a very nice slide where you also see accumulation of these large lipid-rich foam cell macrophages in early atherosclerotic plaques. And we asked the question, what are the metabolic and inflammatory characteristics of macrophage-derived foam cells? And we used the classical model that was originally published by Chris Glass, where we took LDL receptor knockout mice, we fed them a high-fat diet or a normal fat diet for 8 weeks, isolated peritoneal macrophages, and then you end up, in the case of the non-fat diet, with normal peritoneal macrophages, while in the mice that have received the high-fat diet, you end up with foam cells. And we started characterizing the metabolic characteristics of these cells. We performed seahorse analysis, and with seahorse analysis, you can quantify glycolytic rate and mitochondrial respiration of macrophages in the 2 subsets. And basically, this did not show clear differences. So in this graph, you see the extracellular acidification rate which is a marker, or which is a process for glycolysis, while on the y-axis you see oxygen consumption rate, which is a marker for mitochondrial respiration. And as you can see, the normal fat diet macrophages and the high fat diet macrophages map closely together. And also basal glycolysis or basal respiration, if you plot it separately, were similar. So no clear differences there. If we then looked into a bit more detail, we started stressing the cells and looked at their maximal mitochondrial respiration. There seemed to be a minor difference between the two populations. Again, here's seahorse data. And if we stress the system and look at them, the maximum capacity that these cells have to do to rely on mitochondria, the maximum mitochondrial respiration was a bit reduced in the high-fat diet macrophages, and also the spare respiratory capacity was reduced. This was mainly caused by a reduced number of macrophages, because if we would track the number of the number of mitochondria by mitotracker, for instance, or by quantifying mitochondrial DNA, both were reduced. So high-fat diet macrophages seemed to contain a bit less mitochondria. Then we furthered the metabolic characterization, and we performed metabolomics on these cells to characterize 63 different intracellular energy metabolism linked metabolites. And one of the first things that we did is that we made PLS plots of it, a sort of PCA plot where you can define the differences between metabolic characteristics of cells, metabolic characteristics of macrophages. And we looked at, so here you see the populations of cells from the normal fat diet mice. Here you see the cells from the high fat diet mice. And you can then examine which are the key metabolites that discriminate these two populations. And interestingly, if you look at the top list over here, you can see that itaconate was one of the major hits that we had in these comparisons. And itaconate is particularly interesting because it had already been linked to macrophage phenotype. So in the foam cells, we saw reduced mitochondrial mass, we saw reduced maximal respiration, and we saw differences in metabolites, and we were particularly interested in itaconate, which was reduced by the high-fat diet. Itaconate was of interest because, as I said, it had already been published by several groups, including the group of Luke O'Neill, as an anti-inflammatory mediator. It is particularly strongly induced if you activate macrophages, and it sort of provides a negative feedback loop to suppress macrophage activation. And because itaconate was reduced in our macrophages, as said before, this may lead to hyperactivatable macrophages, if this was an important modulator of the phenotype that we saw. So the next step that we did is that we took these macrophages and we LPS activated them to see whether they were indeed hyperactivatable. But not completely unsurprising, that is not what we saw, because what we saw was that these foam cells actually were less activatable. So here you see several of the classical marker genes for macrophage activation. On the left here, without LPS activation, on the right with LPS activation, and you see that several of the markers, IL-6, nitric oxide, superoxide production, were indeed reduced. And also, and this was confirmed by doing RNA sequencing on these macrophages. And this was not completely unexpected, because upon description of the model, Chris Glass had already published that these macrophages were less activatable by the fact that they were accumulating desmosterol. And desmosterol is an LXR ligand, and LXR has been shown to exert anti-inflammatory effects in macrophages. So this fitted with previous findings. But we continued our metabolic characterization of these macrophage foam cells. So we next looked at glycolytic rate, because suppressed macrophage activation would, based on the classical hypothesis that glycolysis is important in driving macrophage activation, so less activated macrophages would be expected to be linked to less glycolysis. But this is not what we see. This is not what we saw, because if we would look at glycolysis, again measured by seahorse assays, either in response to an acute LPS stimulus or in response to long-term LPS activation, there were no clear differences. And also, if we would look at fluorescently labeled uptake of glucose, there was no difference. So glucose consumption and usage in the glycolysis was not different between cell types. And also, if we would look at the metabolic, the intermediates of the TCA cycle, there were no clear differences between normal fat diet and high fat diet macrophages. But to further look into the metabolic characteristics of these differently activated macrophages, we now perform metabolic profiling not on the control macrophages, but on the LPS stimulated macrophages. And here you see the PLS plots of the LPS activated macrophages, again separating very nicely. And here you see the main determinants of these differences. And what immediately caught our attention, indicated here by the arrows, is that there were several metabolites that were intermediates of the pentose phosphate pathway. And the pentose phosphate pathway has been previously shown to be activated upon macrophage activation. Also, when looking at the metabolites in more detail, you can see that different of these pentose phosphate pathway intermediates are reduced in the high fat diet macrophages compared to the normal fat diet macrophages upon LPS activation. Looking into more detail into the regulatory mechanisms of the pentose phosphate pathway and focusing on the enzymes that control the PPP, we could also show that two key enzymes were also repressed in the high fat diet macrophages in the foam cells. And this sort of summarizes these findings of the pentose phosphate pathway. This is the pathway that starts with glucose 6-phosphate and then converts this through these different enzymes into different metabolites that provide precursors that are necessary as building blocks in response to cellular activation. So to demonstrate that the PPP is indeed important for LPS-induced inflammatory macrophage activation, we use two chemical inhibitors that inhibit the first step in the PPP, shown over here. And we could show that if you add these two macrophages that are activated by LPS, you suppress the production of these inflammatory macrophages, and in this case, IL-6, TNF, and nitric oxide. So we see an attenuated LPS response. We see no general inhibition of macrophage activation. I did not show you the data, but for instance, IL-10 and IL-4 responses are completely intact in these macrophages. No major differences in glycolysis or TCA, but reduced levels of PPP metabolites and regulatory genes of the PPP. And how is this hypercholesterolemia or the lipid accumulation in these macrophages mediating this? So it could be that LXR activation, as was published by Chris Glass, is acting also on the PPP and thereby is exerting its effect on this PPP and impacting on the phenotype. So one of the experiments that we did is that we added an LXR agonist to these macrophages and then looked at several of the PPP intermediates. But our main conclusion was that this was not different between normal cells, and, that this was not different between LXR-treated cells or normal cells. So we don't believe that LXR activation is impacting directly on the PPP. To better define what is then actually causing the phenotype, the metabolic phenotype that we do see, we again performed RNA sequencing analysis, and upon pathway analysis of the regulated pathways, one of the key pathways that was the top hit was actually NRF2 mediated oxidative stress response, indicated here by the arrow, and on the right you see a series of NRF2 mediated regulated genes, either in red columns or red labeled columns in the normal cells, or in the orange labeled columns in the foam cells. And you see that the majority of these genes are suppressed in the foam cells. Also, if we would look at protein levels of NRF2 itself, we could demonstrate that the high-fat diet would suppress NRF2 levels, which are tightly regulated in cells, were reduced in the absence, or in the foam cells in the presence of the high-fat diet. To further dive into this, we started working with NRF2 knockout mice together with Luke O'Neill, and we used some of the publicly available data sets that we could find. And what we could indeed then show is that if we take wild-type macrophages and if we take NRF2 knockout macrophages and we look at the regulatory enzymes of the PPP, we could indeed see that they were downregulated in NRF2 knockout macrophages. And what we could also show using publicly available ChIP-seq data is that one of the key enzymes that is regulating the PPP, PGD, actually contains binding sites. The promoter region contains binding sites for NRF2, suggesting a regulatory role for NRF2. And if we would knock out NRF2, indeed we saw suppression of expression of PGD. You see that here under normal conditions, and over here you see that in LPS activated conditions. And finally, if we would measure metabolites of the PPP, they were also suppressed in the absence of NRF2. So to summarize this part of my talk, what we could show is that high cholesterol induces foam cell formation, and that in addition to the previously published mechanism by Grace Class, where desmosterol leads to LXR activation and this suppresses macrophage activation, foam cell formation also impacts on NRF2 that subsequently regulates the PPP, suppresses the PPP, and thereby also impacts on the inflammatory macrophage responses. And based on our experiments, we think that these are two independently operating mechanisms regulating the suppressed macrophage activation of foam cells. So for the second part of my talk, and this is a bit shorter story and still ongoing, I want to go back to atherosclerosis, and this slide from Ira Tebas again illustrates the macrophage and what it does in atherosclerosis, and particularly the contribution to activation, efferocytosis, apoptosis, necrotic core formation, and they can also impact on the collagenous-rich fibrotic cap. We started to work on an enzyme that is particularly important in generation of cytoplasmic acetyl-CoA, and this enzyme is called ATP citrate lyase. It converts citrate into acetyl-CoA, and this then provides precursors, and then this is the precursor for fatty acid synthesis, cholesterol synthesis, and histone acetylation. And we were interested because we saw that macrophage activation coincided with activation of this enzyme. So in Western blots, we could show that LPS led to phosphorylation of ACLY without impacting on total levels, but by increasing specifically phosphorylation of this enzyme. And we could also demonstrate in human atherosclerotic plaques that a portion of the macrophages that you find in these lesions are actually positive for phosphorylated ACLY, also indicating a potential role in these cells. So we generated ACLY deficient mice with ACLY deficient macrophages, performed a bone marrow transplantation, and then did an atherosclerosis study to study its impact on atherosclerosis, to first have a functional readout of the relevance of this enzyme in macrophages in the context of atherosclerosis. And the initial result was a bit disappointing because total plaque area was not impacted by ACLY deficiency. So here you see total lesion area in wild-type macrophages, in mice carrying wild-type macrophages or mice carrying ACLY deficient macrophages, and there was no difference. But further examination actually led to interesting differences, because we could show that necrotic core formation was reduced in these macrophages, and collagen deposition, as measured by total collagen content of minimal cap thickness, was indeed increased in these plaques, suggesting a more stable plaque phenotype. And this increased collagen deposition also coincided with increased expression of TGF-beta, a main driver of fibrosis in plaques. What surprised us a little bit is that if we then looked at macrophage activation in vitro, and we would take wild-type and knockout macrophages and activate them with LPS, these macrophages turned out to be hyperactivatable. So they showed increased production of a lot of inflammatory mediators. And here you see some of them, IL-6, NOS2, TNF, and CCL2. But many of them were increased. So these are hyperinflammatory macrophages coinciding with a more stable plaque phenotype. And if we would look at some of these inflammatory mediators in the atherosclerotic plaques and quantify the gene expression in the lesions itself, they were also increased. So IL-1 beta was increased, TNF was increased, as was CCL2. RNA sequencing analysis and subsequent pathway analysis showed that the main impact that we had of deleting ACLY in the macrophages was on cholesterol biosynthesis. So first we focused on this pathway a bit more. And here you see some of the genes that are in this pathway. Cholesterol biosynthesis seem to be strongly upregulated. Here you see wild-type macrophages and ACLY knockout macrophages. Genes that are important for cholesterol import were up, while, for instance, genes that mediate cholesterol export were downregulated. So cholesterol synthesis was upregulated. Cholesterol export, like ABC, G1 and A1, were downregulated. And we saw a particularly interesting gene, this DHCR24, which is important for the conversion of desmosterol to cholesterol, and thereby regulating the previously already mentioned desmosterol, an important LXR ligand and regulator of macrophages. Indeed, what we could show is that the upregulation of this enzyme, DHCR24, which is supposed to lead to lower levels of desmosterol, and this was indeed the case. So we could detect a lower concentration of desmosterol in the macrophages, and this coincided with reduced expression of a whole series of genes that are LXR target genes and that have been shown previously to be driven by intracellular desmosterol levels. Further analysis of these macrophages also showed additional interesting pathways to be regulated, like cell cycle checkpoints and p53 regulation of cell cycle genes associated with apoptosis. So these were the next phenotypes that we tried to monitor. And we characterized the ACLY knockout macrophages, performed apoptosis quantifications, and indeed what we could show was that in the absence of ACLY, these cells had the tendency to show increased apoptosis, while in addition, also linked to cell cycle, we could show when we would stain for beta-galactosidase that ACLY knockout macrophages had a tendency to go more into senescent or growth arrest phenotype. And then finally, to come back to the topic of the previous speaker, we could also show, because that is pretty important for regulating macrophage or for regulating plaque size and plaque growth and plaque characteristics, that absence of the enzyme ACLY also leads to a phenotype of macrophages where they can show increased efferocytosis. And this might be the reason that the increased apoptosis that we see and the changes in senescence are sort of counterbalanced by also having more efferocytosis and leading to a plaque or a lesion size that is similar between wild type and knockout macrophages. So in conclusion, and this is ongoing work, we're still working on the details of these mechanisms. We can knock out ACLY in macrophages. A main phenotype is a deregulated cholesterol biosynthesis that impacts on cholesterol import, for instance, and on receptors that are related to this, but that also may be impacting on efferocytosis. And in addition, we see that the deregulated cholesterol biosynthesis pathways impact on desmosterol. They show reduced levels of desmosterol. This suppresses LXR activation, and this may actually be one of the explanations that we see such hyperactivatable macrophages. And additionally, we see, and we try to link this to LXR activation as well, that there's an impact on apoptotic processes. And the whole repertoire of these changes leads to more stable atherosclerotic plaques. So in conclusion, atherosclerosis shows accumulation of different macrophage populations that associate with clinically relevant regions and that have inflammatory and lipid-laden foam cell characteristics. We show that foam cell formation leads to suppressed capacity for macrophage activation involving reduced pentose phosphate pathway activation. And ACLY, the enzyme that generates acetyl-CoA, seems to be important for controlling macrophage cholesterol metabolism, and this leads to hyperactivating activatable macrophages that surprisingly coincide with more stable atherosclerotic plaques. And this is the group. This is the joint group that Esther and I have in Amsterdam. And the majority of this work was done by a very talented PhD student who worked in my group, Jeroen Baartman, who's now continued as a postdoc in the group of Esther Lutgens. And this work, most of my metabolic work I do in close collaboration with Jan van der Bossen, who was previously a postdoc in my group and who has now started his own group. Thank you very much.
Okay. Thank you, Menno, for this elaborate talk. Thanks, great data. Let's see if there are some questions. I go to the participants. If you have a question, please raise your hand. Let's see. Yeah, Henry, Henry, please. I'll unmute you. Yeah.
Okay. Hi, Menno. This is great. My question is, do you think the ACLY knockout shifted the subpopulation composition of the plaque macrophage, or just generally affected all the macrophage subpopulations? Like,
that's a very good question. I think we see if we look at the in vitro cultures, we see pretty homogeneous effects on these in vitro cultures. But of course, you never know what the heterogeneity in atherosclerotic plaques will be. So for this, we should do single cell sequencing, like you also explained in your talk, and we are starting to do that in some of our other experimental models. But I think for now, we cannot really say whether there's a shift in certain subpopulations that drive the phenotype or whether the whole is changes, but it's an interesting and relevant thing to study.
thank you.
Okay, there are more questions. I don't see any hands anymore, so then I would say thank you very much for listening into this seminar. It was a great seminar. I would like to thank both of the speakers, and then I'll give the word back to Abel.
Okay, I get to say goodbye. Thank you so much for coming today, and we'll see you next Friday.
Great. Thank you, everyone.
Thank you, everyone. Bye.
Thanks, bye.
All right. All right. So